to the lecture on blood vessels. As you can see from the initial slide, this will be broken into 2 recordings, so I'll do a good chunk today and then I'll do the rest of them next week, but feel free to go ahead if you so desire. So the blood vessels make up the delivery system for the heart. So they begin and they end at the heart. The arteries, just think A for away, they carry blood away from the heart, and all of the blood in the arteries is oxygenated except for the pulmonary circulation, right? The pulmonary arteries are carrying deoxygenated blood to the lungs to be reoxygenated. And in the fetus, the umbilical vessels, the arteries carry deoxygenated blood. Capillaries contact tissue cells. Their wall is only a cell layer thick, and they are meant for diffusion. So they directly serve cellular needs, and then veins carry blood back towards the heart. And for the most part, veins carry deoxygenated blood, with the exception of the pulmonary veins, which are carrying oxygenated blood back to the left atrium. The arteries and veins have 3 layers to their walls: the tunica intima, the layer that comes in contact with the blood, the tunica media, the muscular layer, and the tunica externa, the most external layer. The lumen is the central blood-containing space, and then in capillaries they have an endothelium with barely their basal lamina. Like I said, the walls are extremely thin because they're meant for diffusion. In terms of the tunics, or the layers, the tunica intima is the innermost wall, so it's endothelial cells that lines all the lumens. In vessels that are larger than 1 millimeter in diameter, a subendothelial connective tissue membrane is present. The tunica media is comprised of smooth muscle and sheets of elastin fibers, and sympathetic vasomotor nerve fibers control their vasoconstriction and its vasodilation. The tunica externa, or also known as the tunica adventitia, has collagen fibers to protect and reinforce, as that is the most external layer. And basically, we want to prevent a vessel blowout. Larger vessels contain what's called a vasa vasorum, which is a blood supply just to nourish that external layer, because it's so thick that the outer layers are actually not in contact enough with the blood in the vessel itself. Okay, and here's just a summary slide. And you see the difference between an elastic artery, a muscular artery, and an arteriole, which is the vessel that will connect the larger arteries into the capillary beds. They kind of act like a toll booth to slow down the traffic, the blood flow, as we go into those lace-like capillaries. And you can see how the composition of elastin, smooth muscle, and fibrous tissue varies between them. Okay, and likewise with veins, as we come out of a capillary bed, just like we go from artery into arteriole into capillary, we come back out into venule and then into the larger veins. And the walls are obviously different, with the noted huge difference between arteries and veins with the lack of musculature in the veins. There still is in the larger veins, but it's not to the same degree. So we start with our elastic or conducting arteries. These are the large, thick-walled arteries that have elastin in all 3 layers or tunics. This is the aorta and all of its major branches. It has a large lumen to offer low resistance. Why? Because we want these elastic or conducting arteries to serve as pressure reservoirs. We basically want them to fill, their walls to expand, and then for there to be a massive recoil as blood is ejected, so that it can be sent all throughout the body, even down to our toes. Then we have the muscular or distributing arteries and arterioles, which are distal to those elastic or conducting arteries and have the job of delivering blood to all the body organs. They have a thick tunica media with more smooth muscle than the other arteries, and they are very active in cases of vasoconstriction because they contain the most smooth muscle. The arterioles are the smallest arteries. They're technically like a subartery, and they lead into capillary beds. Their job is super important in controlling flow, so we we take the blood pressure and drop it pretty dramatically as we go into the capillaries because their walls are so fine and thin that we don't want to blow them to pieces. The capillaries are the microscopic blood vessels. Their walls are just a super thin tunica intima, that's it, just a simple squamous epithelial cell thick. And because of this, there are pericytes, these cells that you see yellow in this scanning electron micrograph, that help stabilize the walls and control permeability when needed. The capillaries are such a small diameter that they can only permit a single red blood cell to pass at a time. In all tissues except for cartilage, epithelia, and the cornea and lens of the eye, we have capillaries. And these function in exchange of gases, oxygen and CO2, exchange of nutrients or delivery of nutrients, and then takeaway of wastes, delivery of hormones, etc. There are three structural types of capillaries: continuous, fenestrated, and sinusoidal. Continuous are abundant in the skin and in our muscles. They contain many tight junctions to really connect tightly adjacent endothelial cells, basically shoulder to shoulder, and intercellular clefts will allow the passage of fluids and small solutes. These are also found in the brain, where we want there to be a very thick or very tight and discerning blood-brain barrier. In fenestrated capillaries, think of the word fenestra as Latin for window. So some of these contain literally windows or little pores. So they're more permeable than the continuous capillaries, and they function in absorption or filtrate formation. So you find them in places like the small intestines, the endocrine glands, and in the kidneys. And then sinusoidal capillaries, or sinusoids, have even fewer tight junctions, larger intercellular clefts, or larger spaces between cells in the wall, and large lumens. They also contain fenestrae, or windows, or pores. And their job is to allow large molecules and actual blood cells themselves to pass between the blood and surrounding tissues. So we find these in places like the liver, the bone marrow, and the spleen, which literally serve as cellular filters and cellular reservoirs. Okay, and here's a depiction of each. Capillary beds are interwoven networks of capillaries that form the microcirculation between arterioles and venules. They consist of two types of vessels: vascular shunts, otherwise known as meta-arterioles, which directly connect the terminal arteriole and the first post-capillary venule, as well as true capillaries. So there are anywhere from 10 to 100 exchange vessels per capillary bed, and they branch off the meta-arteriole or from a terminal arteriole. Okay, so here we have the capillary beds, excuse me, with the meta-arterioles here you see acting as bypass channels and to regulate flow through capillary beds. And we have an arteriovenous bypass here, and you see the little sphincters to regulate blood flow even in the capillary beds themselves. Think about when you get cold, your skin is more pale because those capillary beds close off and contract to conserve heat. Precapillary sphincters regulate blood flow into those true capillaries, and they're regulated by local chemical conditions and vasomotor nerve control. Venules are formed at the distal end of a capillary bed when the capillary beds unite. They're very porous, unlike arterioles, and they allow fluids and white blood cells into tissues. Post-capillary venules consist of endothelium and a few pericytes. Larger venules have 1 or 2 layers of smooth muscle cells. Veins are formed when venules converge. They have thinner walls and larger lumens as compared to their corresponding arteries, and blood pressure overall is lower in veins than in arteries. Veins have a thin tunica media as opposed to the thicker one seen in arteries, and instead a thicker tunica externa consisting of collagen fibers and elastin. Veins are known as capacitance vessels, or blood reservoirs, as opposed to pressure reservoirs. Their job, at any point in time, they can contain up to 65% of the blood supply. So here we can see a micrograph of an artery, and what you see here in the pink is the smooth muscle. So compare the thickness of the smooth muscle with that seen in the vein, right? And the vein has a sort of a larger but floppier lumen. So the artery is meant more as a pressure reservoir, whereas the vein is meant as a capacitance reservoir for blood. Right? At any point in time, the systemic veins and venules contain 60% of the blood, the systemic arteries and arterioles 15%, the pulmonary blood vessels 12%, the heart itself 8%, and the capillaries 5%. Veins have adaptations because they lack the smooth muscle that arteries have. They have adaptations that ensure the return of blood to the heart. One is having large diameter lumens that offer little resistance to flow, and two is having valves that prevent backflow of blood. And we see most abundant, valves being most abundant in the veins of the limbs where blood has to climb against gravity. Venous sinuses are flattened veins with extremely thin walls. An example of one is that coronary sinus that you see on the posterior side of the heart. Vascular anastomoses are interconnections of blood vessels. Arterial anastomoses provide alternate pathways or collateral channels to a given body region. These are common in joints, in abdominal organs, in the brain, and in the heart. If you think about it, they're like fail-safes, right? Because if there's a blockage, we want to know that even at least temporarily, there's another way for the body to get blood. Vascular shunts of capillaries are examples of arteriovenous anastomoses. Anastomoses are more common in the venous system than in the arterial. Blood flow is the volume of blood flowing through a vessel, an organ, or the entire body's circulation in a given period, and it's always measured in milliliters per minute. Blood flow should be equivalent to cardiac output for the entire vascular system, and it's relatively constant when an individual is at rest. It will vary widely through individual organs based on their needs. Blood pressure is the force per unit area exerted by the blood on the wall of a blood vessel. We express this in millimeters of mercury, and it's measured as systemic arterial blood pressure in the large areas near the heart. The pressure gradient provides the driving force that keeps blood moving from higher to lower pressure areas. Resistance, otherwise known as peripheral resistance, is opposition to blood flow. It's a measure of the amount of friction that the blood encounters, and is generally encountered in the peripheral systemic circulation. Three important sources of resistance to flow are blood viscosity or thickness, total blood vessel length, and blood vessel diameter, with blood vessel diameter being the most important. Blood viscosity and blood vessel length in healthy individuals should remain relatively constant. So again, blood viscosity refers to the stickiness or the thickness of the blood that's due to a combination of the formed elements, red blood cells, white blood cells, and platelets, and dissolved plasma proteins. And then blood vessel length, we know the longer the vessel, the greater resistance encountered. Frequent changes can alter peripheral resistance, and it most often targets the diameter of the vessel. So resistance varies inversely with the fourth power of the vessel radius, which basically means if the radius is doubled, the resistance is 1/16 as much. Small diameter arterioles are the major determinants of peripheral resistance. Abrupt changes in diameter of vessels or fatty plaque formation from atherosclerosis can also dramatically increase resistance. What happens is it disrupts the laminar flow, and as blood is coursing through, it's like pinging off of things sticking out of the walls of the vessels, and instead of going in a straight line, the blood starts to swirl and ricochet off the walls, and its path is more circuitous and less efficient. If we look at the relationship between blood flow, blood pressure, and resistance, we see that blood flow, or F, is directly proportional to the blood or hydrostatic pressure gradient. So if there's a larger difference in pressure from point A to point B, then blood flow will be greater. Blood flow is inversely proportional to peripheral resistance. So when we say directly proportional, we mean if the pressure gradient increases, then blood flow also increases. Whereas if resistance increases, blood flow decreases. So blood flow is equal to the change in pressure, or the pressure gradient, divided by the resistance. Resistance is more important in influencing local blood flow because it is easily changed by dynamically altering the blood vessel diameter. The pumping action of the heart generates blood flow. Pressure results when flow is opposed by resistance. Systemic blood pressure is highest in the aorta, coming right off the heart, and declines throughout the pathway. Systemic pressure is technically 0 millimeters of mercury in the right atrium. And we see the steepest drop in systemic blood pressure in the arterioles because we're slowing everything down as they enter the capillaries. Okay, so we see that steep decline as we go from the arterioles into the capillaries here. Arterial blood pressure reflects two factors of the arteries closest to the heart: their elasticity, otherwise known as their compliance or distensibility, and the volume of blood forced into them at any time. Blood pressure near the heart is pulsatile, as it goes systole, diastole, systole, diastole. Systolic pressure is the pressure exerted during ventricular contraction, or systole, whereas diastolic pressure is the amount of pressure when the ventricles are relaxing, so it's the lowest level of arterial pressure. The pulse pressure is the difference between the systolic and the diastolic pressure. Mean arterial pressure, or MAP, is the pressure that propels the blood to the tissues. So it's a little different than pulse pressure because of how long we spend in systole versus diastole. So mean arterial pressure is the diastolic pressure plus 1/3 of the pulse pressure. Pulse pressure and mean arterial pressure both decline with increasing distance from the heart, but mean arterial pressure takes into account the differing lengths of systole versus diastole. Capillary blood pressure ranges from 15 to 35 millimeters of mercury, much, much, much lower than in the arteries. And we said low capillary pressure is desirable. One, because high blood pressure would rupture the fragile single cell walled capillaries. And we also want there to be plenty of time for diffusion and exchange of materials, because capillaries are permeable and they are exchange vessels. Venous blood pressure changes very little during the cardiac cycle. There's a small pressure gradient, only about 15 millimeters of mercury throughout it, and the low pressure is due to the cumulative effects of peripheral resistance. So if we don't have that same amount of pressure in the veins, then how do we get venous blood back to the heart? Three major factors: the respiratory pump, which is the pressure changes that are created just during breathing, which move the blood towards the heart by squeezing the abdominal veins as the thoracic veins expand. The muscular pump, which is the contraction of skeletal muscles that basically milk the blood towards the heart, in conjunction with the valves that are preventing backflow. And then finally, vasoconstriction of the veins when they're under sympathetic control. Maintaining blood pressure requires the cooperation of the heart, the blood vessels, and the kidneys. If you remember from the endocrine system, the kidneys start the whole angiotensin-aldosterone cascade. So this is supervised by the brain. The main factors influencing blood pressure are cardiac output, peripheral resistance, and blood volume. So blood flow is equal to the change in pressure from one part of a bed to another, divided by the peripheral resistance, and the cardiac output is related to the same. So blood pressure is cardiac output times resistance, and cardiac output depends on blood volume. Blood pressure varies directly with cardiac output, peripheral resistance, and blood volume. Changes in any one of these variables the body tries to compensate by changing the other variables so long as it can. Cardiac output is determined by venous return as well as neural and hormonal controls. Resting heart rate is maintained by the cardio-inhibitory center and the parasympathetic vagus nerves, right? We want to be under rest and digest at any time when we don't need to be stressed. Stroke volume is controlled by venous return. During stress, the cardio-acceleratory center kicks in and increases heart rate and stroke volume via sympathetic stimulation. This results in end systolic volume decreasing. We want to get as much blood out as possible, and mean arterial pressure increasing so we make sure that we force that blood everywhere we need to force it and under the appropriate amount of pressure. Okay, and here's another summary slide that you can walk through showing when you put all these together, what is the result. There's both short-term and long-term control of blood pressure. Short-term is the neural and hormonal controls, and these counteract fluctuations in blood pressure by altering peripheral resistance. Long-term control is mediated by the kidneys, and it counteracts fluctuations in blood pressure by altering blood volume. Neural controls of peripheral resistance involve maintaining mean arterial pressure by altering blood vessel diameter, as well as altering blood distribution in response to specific organ demands. Neural controls operate via reflex arcs that involve baro or pressure receptors, vasomotor centers and vasomotor fibers, and vascular smooth muscle. The vasomotor center is a cluster of sympathetic neurons in the medulla that oversee changes in blood vessel diameter. The vasomotor center is part of the cardiovascular center along with the cardiac centers, and these work to maintain vasomotor tone, meaning a moderate, constant level of arterial constriction. We don't want our arteries to become floppy. The vasomotor center receives inputs from the baro or pressure receptors, from chemoreceptors that are detecting levels of oxygen, CO2, and acid, and from higher brain centers. The baroreceptor-initiated reflexes are short-term responses. Baroreceptors are located in the carotid sinuses, in the aortic arch, and in the walls of the large arteries of the neck and thorax. Increased blood pressure stimulates baroreceptors to increase input to the vasomotor center. This causes an inhibition of the vasomotor center, causing arterial dilation and venodilation. Right? If blood pressure is too high, we want to relax everything, and it also stimulates the cardio-inhibitory center. And here you can see a summary of this feedback loop here. Baroreceptors taking part in the carotid sinus reflex are there to protect the blood supply to the brain. We want to prevent the brain from frying. Baroreceptors taking part in the aortic reflex help maintain adequate blood pressure in the systemic circuit and make sure that the rest of the body is protected. Chemoreceptors are located in the carotid sinus, the aortic arch, and the large arteries of the neck. Chemoreceptors, like I said, respond to a rise in CO2, a drop in pH, or a drop in oxygen. An increase in blood pressure via the vasomotor center and the cardio-acceleratory center will result from their stimulation. As far as higher brain control of blood pressure, there are reflexes that regulate blood pressure integrated in the medulla of the brainstem. The higher brain centers, the cerebral cortex and the hypothalamus, can modify blood pressure via relays to the medullary centers. Adrenal medulla hormones, norepinephrine and epinephrine, or noradrenaline and adrenaline, cause generalized vasoconstriction and increased cardiac output. Angiotensin II, generated by the kidneys, release of renin, causes vasoconstriction. Atrial natriuretic peptide, or ANP, causes blood volume and blood pressure to decline, causing generalized vasodilation. Antidiuretic hormone, ADH, otherwise known as vasopressin, causes intense vasoconstriction in cases of extremely low blood pressure. Baroreceptors quickly adapt to chronic high or low blood pressure. Long-term mechanisms step in to control blood pressure by then altering blood volume. The kidneys act directly and indirectly to regulate arterial blood pressure through direct renal mechanisms as well as indirect renal, or the renin-angiotensin aldosterone mechanism. The direct renal mechanism alters blood volume independently of hormones, so increased blood pressure or blood volume causes the kidneys to eliminate more urine, thus trying to reduce blood pressure, as opposed to decreased blood volume or decreased blood pressure causes the kidneys to conserve water to make the blood volume increase and make blood pressure increase. The renin-angiotensin mechanism responds to a drop in arterial blood pressure by having the kidneys stimulated to release renin. Renin then, through cleavage, a series of cleavage of proteins, produces angiotensin II. Angiotensin II is a very potent vasoconstrictor that then stimulates aldosterone secretion. And so angiotensin, just think of the name angio, blood vessel, tensin, contraction. It makes the blood vessels contract and constrict, and then aldosterone works on sodium reabsorption and water retention and decreased urine output.